Working with Peter has been some of the most fun I've had, as well as working with several other folks in this room. I'll highlight some of that shortly. So I am going to do a talk that is, as opposed to being focused on results and where things went well, will be focused on interesting problems where stuff didn't work. My goal is to set up the subsequent discussion at least a little bit. However, my computer has decided not to go into presentation mode. So we'll see how this goes. There we go. Something. So my group, we work at the intersection of statistics and biology, environmental science, and we try to find really challenging problems that force the development of some new approaches. And then we collaborate with very talented groups of statistics and computer science to deliver new solutions, but not to Microsoft products at all. Okay. Try that one more time. Maybe, or just blackness. I just wing this, it's fine. I got nothing. Oh, there we go. Now it's. Okay. Sorry, folks. This is not what we need. I'm mentally cutting slides. We'll stay on time, I promise. Rats, I got nothing. Yeah, it's PowerPoint. Yeah, do you want to, let's do these two things in parallel and we'll see which one works first. Still got spinning wheel of death. Yeah, now that's. Oh, we've got a white screen with a spinning ball. All right. Talk amongst yourselves, folks. Okay, can I email this to an address? The email address in there. We can try from that. Peter now thinks it's. Peter thinks it's nope. Oh my God, is it gonna work? Nope, it's not gonna work. Oops. Maybe. Let's maybe. It doesn't look like it's working on my end. No. No worries. I'm gonna be quick. Quick. It might actually be working. You keep doing that, but I'm gonna pretend like this is gonna work. Okay, so we're now gonna get a very quick talk. And I'm going to start out just by framing the class of problems that I'm going to address. We're going to be talking about problems where you have to do pattern discovery as opposed to pattern recognition. The difference being in pattern recognition, you know what you're looking for. And so the problem reduces to simple prediction, right? You're fitting some deep net to recognize pictures of cats and pictures of cats, whatever. And in pattern discovery, you're generally doing something more complicated. Here's an example from functional genomics we've heard a lot today, where you have a whole bunch of transcriptomics data or gene expression data, and you want to find networks of genes sub-networks of genes that are responsible for driving sub-networks of metabolites or metabolism, right? And that is not a pattern recognition problem, it's a pattern discovery problem, because you got no idea what these sub-networks are or what these metabolites are a priori. This is a pattern you have to discover, right? So that's the general class of problems of interest. Now, broadly, for problems of this type, what we're looking at is we have some sort of data and some sort of prior knowledge. And what we're trying to get to is a mechanism. We're trying to understand a mechanism, how the system works. What we have in between the data and prior knowledge and the discovery of that mechanism is some sort of learning architecture. And that could be a statistical model, could be a GLM, it could be a deep neural network, it could be a random forest, whatever. But the key point is that we are learning, that we are using it to try to get to mechanism. Now, depending on how much data we have, the number of samples, as well as how complex that data is, we have access to very different classes of models to try and solve a problem like this. If we have very little data, we're going to be doing very classical stuff. If we have more data, we might be doing piecewise models, Mars, HAL, things like that, to name some local favorites. If we have a little more, we do ensemble methods, random forest, things like that. And then more, and the models get increasingly complex. And ditto for the overall complexity of data, whether we're trying to solve some simple multi-regression problem, or if we have many, many different modes of data we're trying to interrelate. And there are driving data sets in the biological sciences at every level of complexity here, from trying to understand climate science to synthetic biology, all the way down to clinical biology, right? Phase 1 human trials will always be small data. In a phase 1 trial, you're asking the question, hey, if I actually give a person this drug, will they die? That will always be a small numbers game, right? So biology drives not just one set of these methods, but the entire hierarchy of them. And in that setting, we are going from data sources through some learning machine to some representation of what that machine has learned, some object on which we can do something very like inference. Okay, and now I'll give some real examples of what this looks like. And the hope is that these extracted representations that the machine has learned are sufficiently realistic and sufficiently reflect mechanism that we can do things like design future experiments, control systems, etc. Now, one of my favorite projects that we've done in this area is with Ben Yu, who's here, who'll be talking on the panel in a minute, as well as Sumanta Basu. At the time, he was a joint postdoc. Now he's gone on to Cornell. And iterative random forests was a way of doing sort of inference with random forests, but trying to find features that interact together. Now, everything that a forest knows is encoded in paths from root to leaf in individual trees, right? And the idea behind iterative random forest was a very simple one. It was extract the information, extract the sequence of features that are used in individual trees, run them through random intersection trees, and then identify groups of features that co-occur most frequently. Okay, so very simple idea, but it yields really nice insights because you can identify interacting subsets of features that have a nonlinear structure, regardless of how many features are in them, right? So of any form or order. Now, the way this works in practice, after you've extracted that information, and this is the fast version, I apologize and ask questions if you're curious. After you've extracted that information, you can go back to the trees and ask questions also about the decision rules that were actually learned. And if you extract the decision rules along with the features, you can start to draw things like response surfaces, right? And so this method was developed originally to handle genomics data and to answer questions about gene expression and gene control. But since then, we've applied to lots of different stuff, and I'm going to give a different example now. But this is the kind of information you can extract, right? So a pair of interacting features, as well as the actual function that the forest has learned, because that function is encoded precisely in the splits. You extract all the splits, you ensembleize across them, you get a response surface. For binary classification, on the z-axis, you just have probability, and on the x and y axes, you have the range of the support, the domain of the support. Okay, so here is one of my favorite problems in which we've applied this method. So this is trying to learn the controls of an environmental system. In this case, the environmental system in question is an agronomic system. It's corn and soy cultivation, right? So something outside biomedical science, still bio. In this setting, we gather many, many layers of data, hundreds of layers of data that describe the relationship between a plant and its environment. And the idea is, can you learn to control it? Can you learn to drive toward favorable outcomes? So you fit a random forest model, you identify what are the important features, and then you extract something that looks like this. And this plot shows a relationship between potassium and phosphorus, and it shows a sweet spot that is dependent non-linearly on both features. And the first time I showed this was at a USDA conference, and I was booed. And as many of you know, it's not easy to get booed at an academic conference, but I did it. I did it. And it's because this is in a complete contrast to the previous 50 years of agronomics that treats individual features, especially things like potassium and phosphorus, as independent. But it's not. This has been validated now by third parties. It's exactly how it works. But people hadn't treated it as a data science and control problem. And random forest brought to light very nicely these sorts of nonlinear dependencies everywhere. I had a very clever grad student, or still have actually, he's graduating now this semester. Come on. Carl, there we go. Carl had the very clever idea that we take these individual response surfaces that a random forest has learned and do something like forward-backward regression, and then just build a generalized additive model out of the response surfaces that were extracted from the forest. Okay, so this goes from this data, prior knowledge to mechanism, right? Now we're hoping that those individual response surfaces actually correspond to relevant controls for the system. So in the potassium and phosphorus example, it's not enough to control those two things independently. In fact, they're a single control. You have to use both of them simultaneously. And Carl found that if you just wrapped a generalized additive model around these response surfaces, you had about as much predictive power as the random forest from which they were derived. And most importantly, accuracy up to about experimental error in most of the ways we tried it. And now for the last 3 years, we've been using that to drive fertilizer application on a 1,000-acre research farm in Arkansas. And we are now putting about $20 per acre back in the farmer's pocket. And that's more than 30% gain on their total operation. It's a hugely material gain. And it's precisely from the insights derived from this random forest model applied in this setting. Now, that's cool. Okay, so where did things start to go off the rails? So there is tremendous mechanistic heterogeneity. And I just showed you an additive model that treated things as very homogeneous. This is data from a real clinical trial. I think this one was done by Bayer, sorry, field trial done by Bayer. And on the y-axis, we have yield in bushels per acre. This is delta from control regions. And then on the x-axis are just individual fields ordered from worst to best in terms of the impact of the application. And some fields were down minus 20 bushels per acre, so substantial detriment. And some fields were up 40, hugely beneficial. What you wish you knew, of course, before buying this product is which fields were going to be garbage and which fields were going to be great. And then you could put it in just the right places. So how do you do that? So we took, again, an iterative random forest approach to try and learn controls for this setting, except now, instead of just doing random intersection trees on the features that the forest was using, we did it on the observations that live in the leaf nodes. And so now you have a more complex type of object. You have an object that associates interactions or local models with particular subsets of observations. And that creates, depending on where you slice it, a partition on a field site. So this is 8,000 acres thusly partitioned at a reasonably felicitous cutoff. And what you get now are local models with actually completely distinct support. So you learn that there are different drivers that are principal drivers of productivity in this region, this green region compared to this blue region compared to this pink region. And you can actually control decisions like, okay, this is real data. This amendment here is going to be very beneficial. That amendment's going to be harmful. And then it actually flips over here, right? And we can go further and figure out why this is. And it turns out because this tends to be very high moisture, this tends to be very dry, and that actually a drought treatment doesn't matter. But that actually works, right? You can gain insight into how the local model functions. So that suggests a particular way of thinking about the types of models that are being learned. Now, Peter had a paper, is one of my favorite papers of his, which is local polynomial regression on unknown manifolds. This paper, they showed that the scaling, how much data you need to do polynomial regression is just awful. You know, it's really rough. And the fascinating corollary of that is that if you're able to do regression in really high dimension, there has to be some kind of low dimensional structure, or you'd never be able to do it with finite data, right? That's a tremendously conceptually useful result. Now, for the types of systems we study, we know that hundreds or thousands of features are very important to keep around, yet not locally. When we go that local step to dive down to figure out what are the controls locally, in this case, in a very literal sense of in space, but when we dive down locally, things are much, much simpler. So if I can look at the random forest or whatever type of learning machine and say something about where individual observations, in this case, maybe regions of a field, but in another case, maybe patients, where individual observations are embedded, what's near, what's far. And then I can ask for a collection of patients or a collection of field sites, what does the local model look like? And this is precisely the picture of the field that I showed you. Now, this is, you know, sort of where we would like to go, to be able to go from some global model that does an exquisitely good job of prediction to understanding the local structure and the local controls. And this turns out to be reasonably feasible for random forest models. But how would you do this for a deep net? How would you do this in a reinforcement learning framework, right? That is, you know, incredibly open research. But I think an enormously important direction to answer this question, are there these sorts of local structures, local regression, local whatever, that you can extract and then gain scientifically meaningful insight? Okay, another example where we have fascinating heterogeneity. This story is meant mostly to entertain. Now, SARS-CoV-2 infection fatality rates, that's a map across the US circa January 2020, something like that. Is incredibly heterogeneous, right? Infection fatality rates in New York and Boston were much higher than virtually anywhere else in the country. There were other regions, particularly in the Southeast, where you had little pockets of extremely high mortality. And then there were regions where there was very, very little. And you might ask, what drives that? Why is it so heterogeneous? Can you figure out what the drivers are, the process drivers of infection fatality rate heterogeneity? So, you know, we took a data set which allowed us to estimate IFR, infection fatality rate. I won't go to the details, but basically we grabbed counties after their infection fatality after they'd had enough deaths, for some definition of enough, and then we looked at their trajectory over time. Now, I have a research scientist in my group, Josh Ladow, who is absolutely brilliant, and he came up with a theory that a driver of infection fatality rate heterogeneity may be the environmental microbiome, specifically the household microbiome. When he told me this, I said, Josh, that's the stupidest thing I ever heard. But he was very convincing, and he found some money for it, so we did it. There was previously this thing called the 1,000 home study, where they sampled about 1,100 homes in just about 500 US counties. And they did a sequencing method to figure out what was there in terms of fungi, bacteria, that sort of thing. And now, and you could define derivatives of that, like the Bray-Curtis beta diversity, which will become important in a moment. Bray-Curtis beta diversity just tells you how similar the indoor environment is to the outdoor environment, because we got matched samples. We got one sample in your living room, we got one sample on your patio. Okay. Now, there is an old immunological hypothesis called the old friends hypothesis. And the old friends hypothesis states that it was really important to play around and get covered in the microbes with which our species evolved. Okay. And, you know, so particular microbes. And now Josh formulated this hypothesis, it's entirely his. So indoor, outdoor beta diversity on the x-axis, COVID-19 severity rate on the y. If the indoor was very much like the outdoor, so Bray-Curtis beta diversity low, if the indoor is just like the outdoor, then the upper quantiles of the severity distribution will be very compressed. People won't die. Okay. If beta diversity is very high, meaning the indoor is very, very different than the outdoor, it could be anything. And the reason it could be anything instead of just high, the reason that's this hypothesis, because lots of other factors, right? Socioeconomic, demographic, lots of other factors. But this was his hypothesis. Of course, the null hypothesis, just that it's unstructured. All right. Now, here's what the actual data looks like. going to focus on fungal beta diversity for reasons that an immunologist would be happy to explain. But this is the real data. So this is Bray-Curtis beta diversity on the x-axis. This is COVID-19 mortality rate. This is sort of heat mappy thing. And each of these lines is just simple linear regression fit for the quantiles, right? So it's going quantile by quantile. 90th quantile, 75th, 50th, 25th, all positive slopes, 75th and 90th, really positive slopes. Now you ask, okay, interesting, is it significant? The answer is yes. I won't go into the details, but if you look at the infection fatality rate percentile, and then you look at the slope of the regression line against beta diversity as a function of percentile, it's hugely significant, and the effect size is actually massive. The effect size is on par with the median of the mortality distribution. In fact, the effect size is so strong that we compiled a data set about 745 socioeconomic and environmental factors that influence COVID-19. The only ones that are actually stronger than this fungal microbiome measure, this Bray-Curtis beta diversity, are percent population over the age of 85 and a couple variations of that, percent population of high density housing, percent population in residence over 10 years, mean household income, mean soil moisture levels. And then there's a few others, just variations of this, different breakdowns by ethnicity and things like that. But that's it, right? The effect of fungal is nearly as strong as those things that are very, very obviously very important. And in fact, you can identify just a few fungal genera that appear to drive this pattern. The strongest genera exhibit exquisite patterns. Here on the x-axis for both these plots is indoor relative abundance. This is outdoor relative, or sorry, vice versa. Outdoor relative abundance, indoor relative abundance. Both these plots are reasonably diagonal, meaning that things happen when they are similar, i.e. when the Bray-Curtis beta diversity is close to 0. And the color chart here is mortality suppression. How many it's infections divided by deaths. How suppressive is the presence of this microbe? And for these microbes, the effect sizes are very significant. They're very, very significant. If you have a good amount of this outside and you have a good amount of this inside, yeah, you're going to have, considerably less death in your county. What's also very remarkable is that this pattern and everything I just said, it only holds in counties where residents have been there for 10 plus years, right? So this is a heat map of the US, human population stability, county by county, or at least the 500 counties for which we have data. And then we have existing residents per new resident. So incoming residents per new, or sorry, existing residents who are already there per new incoming residents between 2010 and 2017. Why those years? The years we found data for. And it goes from 4.3 to 250 with a median of 14. If you threshold at the median, so these are high turnover counties and these are low turnover counties, all of the predictive power, all of our capacity to predict as a function of Bray-Curtis beta diversity concentrates in the counties with low population turnover. And from that, you can do nice things like make a risk map of the US, where size of the dot is proportional to mortality suppression. So also there's a nice story about soil pH that goes along with this and why this might be functionally true, but that's cool. So now that's all nice, but the entire story I just told you is dependent on a brilliant bit of intuition by a very, very clever computational ecologist. Could you have got that from the data alone, right? You would hope that you could recover that just through a machine learning process, right? So can you rediscover the old Friends hypothesis from data alone? So we tried IRF, we tried quantile regression forest, we tried distributional random forest, we tried a few other things too that weren't actually random forest. But all the results were very similar. We could recover the really strong main effects like this, because they're super obvious, but we couldn't recover any higher order interactions and nothing was particularly stable. So this is in the regression problem. We have 745 predictors and then the outcome of infection fatality rate. So the hypothesis is that we have too few independent observations of the upper quantiles for any of these methods to nail this problem for us. And I'd be really interested, Peter, to discuss, the distributional, very interested to discuss that with you, show you the results. But it illustrates that these methods are still fairly data hungry, right? 500 data points, not nothing. Still can't resolve it. Okay, one last story that I'm going to very fast. So here was a project that instead of small data being a problem, we had essentially infinite amounts of data because we had access to a simulation. So we're trying to design small molecules to disrupt SARS-CoV-2 proteins. Okay, for some reason, the Department of Energy decided it wanted to compete with pharma. Why it did that, I have no idea, but they gave us a bunch of money to do it, so we did. Now, this is in collaboration with Dan Jacobson's group at Oak Ridge National Laboratory and Marty Head's group, a couple others. In this setting, we have a target protein, and this protein has some binding site that if we could disrupt it, we would prevent the function of the protein. And we're trying to design small molecules that fit in that binding pocket very tightly and constitute essentially suicide inhibitors of the protein. We have a simulation that's okay to say whether or not that a small molecule will satisfy that. We built out a reinforcement learning approach. Very clever student of mine, Yulin Wu, who's a grad student here at Berkeley, got it into ICLRs. Very nice job. A few clever bits, but I'm going to skip over it in the interest of time. The point is that he built a really nice deep learning model that outperformed everything else, including MulDQN from Google, actually pretty handily. These are pretty big differences in terms of delta G for the energy calculation. But then this is the important plot, this unlabeled thing. I don't know if you can see the colors, but it goes from light to dark. And it's unlabeled because it doesn't matter what the labels are. We tried labeling this number of carbon rings. We tried labeling it by other molecular features. The latent space for this wonderful network that performs so beautifully is totally uninterpretable, hideously so. Like if you squint and zoom way in, you can convince yourself that there's sort of some local structure and things partition out locally, but there's absolutely nothing obvious from this. So we fit a method that could design molecules, but what did we learn from it? Design principles for molecules? Certainly not. We learned virtually nothing from this exercise, right? What have we learned from AlphaFold? Where are the new soft matter physics? Where are the new design principles for proteins? Nada, freaking nothing. How do we use these incredibly predictive methods to do science, to gain insight? That's the huge open problem. So in this trajectory from data and prior knowledge through to mechanism, there are still some really big holes. Like we've heard some amazing talks on some of these components down here today. And these are amazing and they're working so well. The methods up here are working better, but they're still very data hungry. And then methods up here, there is nothing. The saliency maps for regions Giles Hooker could tell us at great length are just garbage. And then Lyme, challenging too. So this is the problem, I think, right? How do we do inference in these settings? Okay, I'm going to shut up. Thank you very much, everyone.
Okay, let's take 2 questions.
Just a quick question about the buckets and just a quick question about is that controlled for
Which one?
the fungal exposure?
Yep. Yeah. Age, absolutely. Yes. So let me rephrase that. Can you show that the effect of the fungal microbiome is not dependent on the age of the population? Can anybody count? Yeah, you can. Targeted maximum likelihood, random forest. Yeah.
Okay, another question.
Can you say a bit what deep learning model you used? Actually, we have some interpretation method. Hope you will try.
Okay, we're excited about that. So it's a graph policy network. The innovations for this, a lot of it is pretty similar to MulDQN and stuff that was done before. The innovations are less in the reinforcement learning side and more on the science side. So in most of the folks who've tried to do this molecular design stuff do it atom by atom. That's how they phrase the Markov decision process, atom by atom. Instead of that, we took a library of synthetically accessible fragments and actual chemical reactions that humans know how to do, about 4 billion of them. And we use that as the input. So we explored a design space so our molecules were always real and we always had a path to constructing them. And then we had a slightly clever way of doing really curiosity-driven exploration. But other than that, it's just a deep graph net, convolutional graph net, optimized by proximal policy optimization. I'd love to talk to you, Ben, about how to gain some insight. And maybe, you know, presumably there's something, certainly for something as exquisitely predictive as AlphaFold, there's something to be learned if we but knew how to query.
Input as continuous features.
The input are a discrete molecule, a graph, right? So it eats a graph, and then every graph can be associated with a measure of quality, and that measure of quality can be computed by simulation. So the reinforcement learning, every time the reinforcement learning algorithm wants another data point on which to train, it can just call it and ask for it. Thank you very much.
Okay, let's take our speaker again. Time for coffee. Let's come back at 3:30.